Hi everyone. So my talk is about finding three principal gradients of gene expression in the human cortex, these three patterns up here, and what they mean. So many of you will be familiar with the Allen Human Brain Atlas, which is this data set that sampled gene expression at over 3,000 regions across the human brain, about 2,000 in the cortex. And one of the things you can do with these data is you can run PCA, principal component analysis, on the region by gene expression matrix and look at those principal components. So why would you want to do that? Well, one reason is that when you look at the first principal component of the Allen Brain Atlas gene expression matrix, it seems to be something that's quite relevant for many different brain functions. In particular, this PC1 was shown by Burt and others to be very closely indexing the T1 weighted T2 weighted ratio from MRI, which itself is an index of gray matter myelination. And it's also associated with disorder gene sets and other properties related to anatomical hierarchy. So that raises the question of what about other principal components of this gene expression matrix? Maybe there's more than one meaningful gene expression gradient in the human brain. But the challenge with looking at higher order gradients is that this is the only data set that we have, the Allen Human Brain Atlas, and it comes from only 6 brains, which brings the challenge of how do we know that when we compute higher order principal components, that they are reflecting something generalizable about the human population versus just something specific about these 6 brains. I mean, you can compute as many as you like, but that doesn't mean that they're meaningful, or specifically whether they're generalizable. So in our project, we looked at defining a metric for generalizability using the 6 brains. And the way we did that was splitting the 6 brains that we have into 2 sets of disjoint triplets and processing those completely independently to get principal components, principal gradients, and then taking the absolute correlation between those sets of gradients, which came from completely different independent data sets of 3 brains. Then repeating this for all 10 possible ways that you can split the 6 brains into disjoint triplets. The idea being that if we see the same result or a similar enough result between any of the possible splits of these 6 brains, that would suggest that if we were to get another 3 brains from the wild, so to speak, we would probably also get the same result. In other words, that the result we're finding is generalizable. So here on the right, you see the results of doing this for the first 3 principal components. The first component, each of the dots here is a comparison between one of the ways of splitting the 6 brains into disjoint triplets. And you can see that for the first principal component, most of the 10 pairs are highly correlated around 0.7. But for PC2 and PC3, they're not very correlated at all, only about 0.1. And when you plot them on the brain, they look much noisier and not like this smooth gradient that we see for PC1. So maybe PC1 is all there is. However, this result was computed using all of the genes that exceed the background noise in the Allen Human Brain Atlas. And other studies have shown that not all of these genes are equally stable. And you can measure that stability just by taking every possible pair of single donor brains and calculating the correlation of the gene's spatial expression between those donors and averaging across every pair, a metric called differential stability. So we can rank all the genes by how stable their correlation is between pairs of donors in the data set. And then what a lot of studies do is filter for only the most stable top 20%, 10%, 5% of genes when doing analysis, because maybe the others are too noisy. And so when you do that, you can make a plot something like this. So the first column here is the same data as on the left here, but compressed into a single column. So red, purple, and blue for PC1, PC2, and PC3. And then at each point on the x-axis, we're filtering out the noisier genes. So here we filtered out 50% of the genes, and here, right on the right, we're retaining only the top 10% most stable genes. And you can see as we filter more genes, the correlation between the triplets becomes higher. And indeed, at 10% of genes, it looks like maybe PC2 is now also something generalizable that would apply to the whole population. We're getting about a 0.7 median correlation between all the different pairs of triplets. PC3 still doesn't look that good, though. It's below 50%. And also, when you look at those gradients using just the top 10% of genes on the brain, PC2 now looks much smoother and looks like something that might be worthy of analysis. But retaining only 10% genes isn't a lot. I mean, that's about 1,500 genes, and you can't do enrichment analysis very easily on that. So can we do any better? Well, one idea, so here's the same two plots from before. And one idea is that not all of the regions, here we always using the Glasser pastillation, HCP, MMP. Not all of the regions have the same number of the 6 donors with samples falling in that region because the regions are quite small. And that suggests that we might have a problem whereby the regions that sample fewer donors have a row in the gene expression matrix where we cannot distinguish generalized spatial variation of interest from whatever variation is specific to just this individual donor brain in the dataset. So what if we drop those regions? We can drop all the 43 regions that have samples from only 1 or 2 donors, which gives us a bit of a patchier result, but also a result that is much more stable. So you can see here that when we filter for only the 3+ donor regions and then run our PCA, PC3 now has a higher, about a 0.8 median intertriplet correlation with top 10% genes. And even if we go to the top 20% of genes, we still have pretty high correlations, greater than 0.6 for all 3 of the first 3 principal components. PC4 is down here somewhere, so we're not showing it. So that's better. Now we're retaining top 20% of genes and getting a generalizable result. But what can we do better still? Well, one way is to change from PCA to diffusion mapping, which is a nonlinear dimension reduction technique. And interestingly, when we take the same data but use DM rather than PCA, we can now go all the way to 50% of genes while still meeting this criterion of PC3 having a greater intertriplet correlation than 0.6. So that's really good. Now we've got about 8,000 genes to do analysis on, and we have these 3 components that look quite interesting. And these are all very highly correlated above 0.85 with each other, whether using PCA or DM. So we are getting the same result, just DM is extracting more signal from the noise. And the next thing that we found was, okay, now that we see that DM is better, what if we didn't want to have these very patchy maps that are missing about 40 of the regions? Well, with DM, if we go back to using all of the regions, yes, all the intertriplet correlations get worse, but not that much worse. And now we, by the same criterion of median above 0.6, we can now keep just 20% of the genes, but have all the regions and get these really nice smooth maps, which would be great for spatial analysis. So in summary, when filtering the Allen Human Brain Atlas to look at higher order gradients of gene expression, we find that there's a trade-off between filtering genes and filtering regions, which allows us to look at 2 higher order gradients that seem generalizable. And the trade-off is we can either have all the regions, but only 20% of genes, this option here, or we can have about 2/3 of the regions, but 50% of genes. And both of these are equivalent. Again, these 2 sets of gradients in the matching regions are correlated above 0.85. So do these gradients actually mean anything? Well, the first thing was we looked at the smooth maps and asked if they're similar to any of important imaging maps that are commonly studied in the literature. So here's a set of imaging maps that was published in various reviews. And we see that, as was found previously, the T1 weighted, T2 weighted ratio, which indexes myelination, as well as cortical thickness and the oxygen metabolic rate are all very strongly correlated with the first principal gradient. But interestingly, aerobic glycolysis, as measured by the glycolytic index, as well as developmental expansion and evolutionary expansion, are much more correlated with the second principal gradient than the first principal gradient. So this is strongly suggesting that even though these maps kind of have a rostral-caudal gradient on them, they don't all align to the same underlying gene expression gradient in the brain. There's at least 2 different gradients that are driving a lot of the imaging phenotypes that we observe. And another way to show this is in a multivariate way, by just taking this bunch of imaging maps and running PCA on them. And indeed, the first principal component of this bunch of maps and various subsets of this is actually the second gradient of gene expression. And that's strongly correlated above 0.6, with the second gradient of MRI that's being the first gradient. So that's one interesting result. But what are these G1 and G2 and G3? What do they actually mean biologically? So we can calculate gene weights from each of these maps by taking the 50% of genes and looking at how correlated the patterns of those genes are with the individual genes with the map. And then when we use gene ontology enrichment on that set of gene weights, we find that G2 is a gradient that is strongly enriched in the positive end for metabolism, metabolites and energy, metabolic process, and at the other end for epigenetics, whereas G3 is enriched in the positive end for inflammatory response, as well as blood vessel development, immune processes, and synaptic processes at the other end. And we can confirm that by looking at the enrichments for cell types, where we find for G3 in particular, a strong negative enrichment for synapses, neurons, and a positive enrichment for all the glial cells. So it looks like there's something going on here with G3 being a neuron versus glia gradient in the brain. Another way to understand what these gradients are actually doing is developmentally. And the way we did this was taking the same set of gene weights and taking a dot product with another gene expression data set that only has 11 regions, but has them at different stages of development. And then we can look at whether the pattern of the gene expression in those 11 regions, in these 11 regions like this, is similar to the same 11 regions in the original Allen Human Brain Atlas. What we find is that in the adult brains above 18 years old, all three gradients are very strongly correlated between these two data sets. So that's a kind of validation, not a full validation, but it's great because this data set, BrainSpan, is RNAseq and the original AHBA is microarray. But then what we can do is look back in time at the pre-adolescent and prenatal brains. And here we see that G1 and G2 were fairly consistent, even prenatally, but G3 seems to only emerge after adolescence, which kind of aligns with the idea of it being an immune-related gradient, because the immune system changes so much in adolescent development. And what's super interesting about that is if we then take a disorder data set from imaging maps here, this is cortical volume changes in 3 disorders, we find that autism spectrum disorder seems spatially associated with both the first and second gradients of gene expression, whereas schizophrenia is associated with the third gradient. And that makes sense because autism is diagnosable early in development, and G1 and G2 are stable early in development, whereas G3 emerges in adolescence, which is also when schizophrenia becomes diagnosable. So that's all our results so far. And in summary, there are 3 takeaways. The first is that it's likely that there are at least three gradients of gene expression in the human cortex, not just one. And to find these in the Allen Human Brain Atlas, we need to filter both genes and regions and use diffusion mapping to better extract the signal from knots. The second gradient is metabolic, and the third is related to immune processes and glia. Second takeaway is that not all imaging maps align to a single gene expression gradient. In particular, aerobic glycolysis, developmental expansion, and some other maps align with G2, not with G1. And thirdly, these 3 gradients are linked to neurodevelopmental disorders. G1 and G2 exist early in development and match with autism spectrum disorder, whereas G3 emerges in adolescence and matches with schizophrenia. So that's all our results so far. The paper should be being submitted soon. If you have any questions, feel free to write to me here. And thank you very much for listening, as well as to my supervisors, Dr. Petra Berters and Prof. Ed Bullmore, and to my funders at the University of Cambridge. Thanks.